Okay, are there any questions? If there's no questions, I'm going to begin. Everything's good, huh? I somehow question that. Alright, let's take a look at where we're at. Come on, share the screen. There we go. So you should be seeing my screen now. We're going to be discussing Lab 7, throat and urine cultures. And hopefully you will be working on your unknown work, your unknown project. I've got to get that emailed out to you. But I want to discuss and start the unknown project. So we do need to talk about that as well. Any questions about what we're doing? All right. So let me go ahead and close this. And I think I want to talk about the unknown instructions first. And the unknown project. Hopefully tonight I will email you the information you need to get started on your project. Meaning I will email you a virtual bacteria. And then all term long, the rest of the term, you will be attempting to identify it. Why isn't this opening? Ah, there we go. So this paper tells you the project, the identification of a bacteria. You're not going to get a mixed culture. I guess I should change that because we decided it's too difficult doing this as a virtual experiment to do it as a mixed culture. And let me see if that's right also. Does anyone remember how many points this project is worth? It might only be worth 75 points. Let me take a look here. Always check the syllabus. Yeah, the identification of the unknown project is only worth 75 points. So I need to update this. This is an older version. When this is something that goes back before COVID-19. Anyway, so there's the introduction and rationale for the project. You can read that yourself. The project format. You're going to isolate a bacteria species. Write down daily lab notes. And you know I need to show you that. I have a place for you to write down your daily lab notes. Come on. And use your daily lab notes to write up a hard copy, which obviously you gotta email it to me, but you can type it up, and then identify your unknown. The report should be written using bullet points, not an essay style. Please use 12-point font. My eyesight isn't very good anymore, and I wear glasses, so please use 12-point font. You do not need to double space. You should have descriptions of the tests you ran, the information you hope to gain from these tests, the observations you made from the tests, and your conclusions. And I'll talk a little bit more about that. So observations are different from a conclusion. Don't omit data to make your project look better. Drawings and photos and tables are very helpful, but they aren't required, with the exception of one table or chart at the end. I'll talk about that. Each report entry must have a date and a time. If you only did one experiment in one day, you only need one date and one time. Occasionally, students would have more than one report in a day, in which case they should date and time each experiment. The thoughts and words should be your own report. You may get advice from other people, but write it in your own words. It is mandatory that your report include a table outlining the process you use to identify your unknown organism, and I'll show you an example. You should also, at the end of the report, have a microorganism paragraph, meaning you've identified your bacteria. Now, in a paragraph, tell me about that bacteria. It only needs to be a paragraph or two in length. Do not exceed one type page for this section. And then on top of that, you should have a literature citation page where you formally cite the literature, and you can use whatever citation form you want, MLA or APA or some other. Just be consistent, meaning always use APA or always use MLA. So here's an example of your table in your report. And you could have a flow chart. It doesn't have to be a table like this. You could have the possible species. Let me get this in there. Are you guys seeing this? Hello?
Yes.
Okay. So this would be the possible species, gram-positive coccus. And then you run the catalase test, and it narrows down these species to these 4, which are catalase positive, and then you do the PR mannitol, and it throws out Staphylococcus aureus, but you still have these 3. Then you do PR glucose, and you have these 2 in the running, and then you do citrate, and only Micrococcus varians is now under consideration. So you've identified your unknown species. Let me state, you could do one more test to confirm that your unknown is Micrococcus varians. It's not required, and in this case, I don't have it shown. All right, any question? You do need to have a table in your report near the end telling me how you obtain your unknown bacteria. And it was by these, 5 tests here. Plagiarism, please read about that. Obviously, plagiarism will not be tolerated. There is a grading criteria. I'll show you that in a little bit more detail. You can read about it here, but I'm going to show you the rubric I'll use for grading the paper. All right, that's about it. Where's my rubric for grading the paper? I think it's under week 5 or week 6 lab. Week 5 lab. All right. So there's the unknown instructions. Oh, here's something I forgot to mention. You do need to use dichotomous logic so that when you run a test, let me see if I can open that. That you have one of 2 choices. So you can do the gram stain, and it's either gram positive or gram negative. You know it's gram positive, so you throw out the gram negatives, and then you do your next test, which could be something else. The point is that the possible species, we do the grams test, and there's 4 possible species, and then you do a next test, and it narrows it down to 2 and to 2, the rectangular and the non-rectangular cells. And then we have it's rectangular, and then you find that the colonies are not white or white. Okay, do you see dichotomous logic? Any questions about dichotomous logic? You must use dichotomous logic. Essentially, that means you'll be running one test at a time, and you'll be waiting for the test results before you decide which test to run next. And that will be forcing you to use dichotomous logic. Let me briefly show you this. This is the list of the possible bacteria species you'll be working with. Let me download it because it'll show better. So on the left are the possible bacteria species. I don't know, there's maybe 30 of them. And you'll note that the top half are gram negative and the bottom half are gram positive. And then you can divide them by, oh, I don't know, what would be a good one? PR lactose. All of these top 4 are AG and the PR lactose, and then these bottom ones are negative in PR lactose. I only chose that because it's easy to talk about it. Anyways, if you do the gram stain first, and you don't actually get a choice in this matter, you would then split it down to this part of the chart or that part of the chart, and then you do PR lactose. If it's gram negative, you'd split it down into these or those. Everyone see dichotomous logic? Any questions about that? If not, I'm going to close this. There's a sample journal, and let me show it to you. It is an example of your written journal. Obviously, this was written, I think it's over 10 years ago, by a professor. And you will want to be similar, although you won't start out as he does, because he's starting with a mixed culture and having to streak it out for colony isolation and then obtain a pure culture. And then start his tests after that. You will already have a pure culture when you get it, and so you will be running tests from the start. But let me point out something that's important. Generally speaking, on a day, say written in 2004, you'll have your observations, which were from the tests that you had ran previously and were due this day. An observation is an observation, what you're observing. These will be the results of the test. And then the activities, these will be the tests that you're going to run today after you've made your observations. And then, he doesn't have it here, but the next thing you do would be the conclusions for your observations. And of course, there's no conclusions because there's no observations. But let me scroll, and I'll show you where he's got it. Here we go. February 12th. Observations. Summary of observations. Activities that he did in the day, and I don't know why he's got the conclusions in with the activity. All right, any questions about any of that? That's what you should do. You should have, generally, on a day, your observations, your activities, what tests you're going to be running that day, and then your conclusions. And it doesn't have to be then in that order. You could put observations, conclusions, and then your activities. Remember, an observation is what you observe, the test results. The conclusions are your interpretations on that result. So you look at the gram stain and you see purple cells. That's your observation. The conclusion is that the cells are gram positive. Everyone notice the difference? All right.
I had a question. How exactly will we conduct the experiment, considering we won't actually do the experiments?
What you're going to do is, I'm going to email you a number which will stand for a bacteria, and then I will email you some test results, and initially I'm going to choose which test you run, but later on you're going to choose which test you run. And the reason for that is I figured out that this project would involve 100 or more emails on my part. And I just said that's too much. But if I choose the first 3 tests you're running, instead of every student getting 3 emails, they're getting 1. And in actuality, I'm going to send the same one to everybody. It's going to have everybody's results. And I just reduced the number of emails by, let's see, that's 20 students times 3. I just reduced the number of emails by 60. So I'm going to do the first 3 tests for you. And then you're going to look at your results and observations, make your conclusions, and then determine which tests you want to run next. Okay? And then you ask for it, and you can find how long that will take. When you ask for it, I will incubate it. Like you're asking for the gelatin hydrolysis test, take a look how long it takes. This one runs for a week before you get the results. And after a week of incubation, I will send you the results. You have to make your interpretations, the conclusions, from those results.
I thought you said due May 15th.
Yes? No, the assignment isn't due May 15th. I think that's the infectious disease project.
Okay.
Okay. This project, the unknown project, will be due a week before finals week.
Okay.
Okay? So it will take you time to get the unknown project done. So don't wait to the end. All right? Like I said, when you request a test, some of them, like a catalase test, if you have the cells available, I can send it the same day. But bioesculin, that will take 2 days to run, and gelatin hydrolysis takes a week to run. And so if you ask for gelatin hydrolysis, you're not going to get your results for a week. Everybody clear on that?
Yes.
All right. Now where was I? I talked about the sample journal. Oh, I want to talk about the grading. There it is. How the report's going to be graded. Now there are some sections that are required. Obviously you have to have the body, you have to have the in-text citation, you have to have the formal citations, you have to have a paragraph discussing the bacteria, and you have to have a table going through how you identified your unknown bacteria. So all of those are required, but the grading will largely be the utilization of resources. Did you use excess media, more media than you needed to identify your unknown? Was dichotomous logic used in selecting your tests? Were all relevant tests used? Like I once had a student who, for some reason, she decided she wanted to run every single test, even if it wasn't relevant. And so I had to run through her report and say, you ran this test, but it didn't help you, in which case she was penalized. Were the correct species identified at each step? Like, for example, you'll be given the gram stain very early. So you should someplace state that my unknown species, at the very least, is gram positive or gram negative. And it would be a good thing, although you don't have to state it, you could state each species. There's about 15 of each. Okay? But you should, at the very least, say my unknown is either gram positive or gram negative of the possible species. The completeness of the report. Is there any instance of missing information? Effective and clear communication. Is your English, your grammar, your spelling, and your punctuation good? If it's excellent, full points. If there's lots of errors and the paper is hard to read, you'll only get 1 point out of this. Nobody has ever turned in a paper where I gave them 1 point. The species identified. If you got the correct species, you get 10 points. Within the same group or type, you get 7 points. If it's outside the group, like outside the gram stain, you only get outside the group, but the gram stain is correct, you get 5 points. If the gram stain is incorrect, you get 0 points. And that'll be a total of 75 points. All right, any questions about the grading? I think we can shut the sample journal. Like I said, take a look at these, the sample journal, obviously instructions you should have looked at already, how to follow a dichotomous game plan. This is the list of the possible species. You'll need to look at this list to narrow down your unknown and then decide which test to run next. And then lastly, you need to write down your unknown notes. And I've set up a page for doing that called the Unknown Notes. When you open it, I don't have it because I'm an instructor, but on the right, you'll have a link here for detailing your results. And each day, you have a test result that you want to write about. You submit a paper. Okay? And you can have them all linked together, so you just add to it, or you can have them independent. I don't care. The point is, write down your unknown notes here so that I can reference it if I need to. The very first thing I'm going to do is send you the number of your bacteria. You need to put down that number in your unknown notes, because if you come on your unknown notes, although I'll send you the first test results, if you don't have your number and you ask for, I would like to do, I don't know, glucose fermentation, I will say, well, that's good, I would like to send that to you, but in your unknown notes you don't have your unknown number, so I don't know what results to send you. So you need to write down your unknown number. And then put down your results, your interpretation, what your possible species are, and then what future tests you'd like to run and why you'd like to run them. Please submit all of that information in a document or paper. Do not put comments over here, because I'm going to be giving you comments. And by the end of the term, you'll have a whole bunch of comments from me. So you are not supposed to write anything in the comments. Put yours as a document that you submit. Any questions about that? All right. I think that's it for the unknown project. I will try and send that out tonight. It'll have to be after the lab. Because I'm a little bit behind schedule. I don't have that ready, but I'll get it ready tonight. Let's go on to lab 7. Okay, lab 7 covers throat and urine cultures. There is some reading that you should do before you begin the lab. It's not too bad this time. And there's some supplemental material. You'll be watching video clips. There is no virtual lab that works for this lab. I put, what do you call that, crossouts, because the virtual labs may become workable in the future, but at the moment they're flash and they won't run. Primary learning objections. Explain how to properly collect throat and urine specimens for routine culture. List common bacterial pathogens isolated from throat and urine cultures. Know the culture media used to cultivate these pathogens. Recognize which tests are used to identify throat and urine pathogens, and understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. Let me put this at the bottom so it'll be less out of the way. Once specimens are received in the lab, they're logged and they're processed in a timely fashion. Specific culture media are inoculated and incubated for throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. From throat, blood agar is the most commonly used media used for isolating throat cultures, largely Streptococcus pyogenes, but chocolate agar is also used to isolate Haemophilus influenzae, which is a bacteria commonly seen in the ear, nose, and throat of children, particularly small children and babies. Streptococcus pyogenes, if you don't know, causes strep throat. For growing urine cultures, blood agar, McConkey's agar, and E and B agar are frequently used. And that's for isolating Streptococcus, Enterococcus, Staphylococcus, many species of Enterobacteriaceae, like E. coli, and then Pseudomonas, which is similar but different from Enterobacteriaceae. Throat cultures. Sore throats can be caused by bacteria, in which case they'll have white patches on the tonsils and the back of the throat, usually caused by Streptococcus pyogenes, also known as group A strep. A picture of that. Strep throat can be determined by swabbing the back of the throat or the tonsils where there's redness and white patches, and then swabbing that culture on a blood agar plate. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy because humans are very lousy, if at all, at throwing off Streptococcus pyogenes. Streptococcus pyogenes is fastidious, and so the culture media has to be supplemented with 5% sheep's red blood, or it's called the blood agar plate. You then observe the plate for small, clear colonies surrounded by a complete zone of hemolysis. This is usually Streptococcus pyogenes. But you only need 2 biochemical tests to definitively prove that the beta hemolysis on a blood agar plate is coming from Streptococcus pyogenes. You run the catalase test and the bacitracin susceptibility test. This is showing you blood agar. And you notice how the bacteria, which is right there, is surrounded by hemolysis. This whole region is white. This is a normal throat microbiota, and this is hemolytic bacteria in the throat of the patient. Catalase is an enzyme produced by most bacteria to break down hydrogen peroxide. Here is hydrogen peroxide. 2 of them, in the presence of catalase, produce 2 water molecules and oxygen. The oxygen creates bubbling, which can be easily seen. Most bacteria that grow in the air produce catalase. However, Streptococcus and Enterococcus are negative for catalase production. So if in the catalase test it looks like this, it could be Streptococcus pyogenes, and if it's positive, it's definitely not Streptococcus pyogenes. Bacitracin susceptibility testing is you swab the entire plate, well, not the entire plate, I guess this region of the plate with bacteria, and then you put on the bacitracin disc, which has the antibiotic bacitracin. If the bacteria are sensitive to bacitracin, then you know you have Streptococcus pyogenes or group A strep. If the bacteria are not sensitive to bacitracin, then it is not Streptococcus pyogenes. It's some other bacteria. That is beta hemolytic on a blood agar plate. Okay? And Streptococcus groups B and C will look like this. Streptococcus pyogenes will look like this, or Streptococcus A. Any question about that? All right. Another organism that's clinically significant and isolated from the throat is Haemophilus influenzae. which is a gram-negative coccobacillus. It's the most common cause of ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is fastidious and will not lyse blood cells, so it will not grow on a blood agar plate, but it will grow on a chocolate agar plate. This is what CAP is. And here you can see Haemophilus influenzae growing on a chocolate agar plate. Curiously, Haemophilus influenzae will not grow on a blood agar plate, but if you streak Staphylococcus aureus down the blood agar plate this way, and then streak Haemophilus influenzae across the streak of Staphylococcus aureus. Let me blow this up a little. The Haemophilus influenzae will not grow in this region of the plate, but near the Staphylococcus aureus, the Haemophilus influenzae will grow. And the reason is that the Staphylococcus aureus is lysing the red blood cells, which is releasing nutrients for the Haemophilus influenzae to grow near the Staphylococcus aureus. But further away from the Staphylococcus aureus, we have no Haemophilus influenzae. Any question about any of that? All right, for urine cultures, they're used to diagnose a urinary tract infection, or U2I. Bacteria can travel up the urethra to the bladder, and females are more predisposed to developing a U2I than males because of their shorter urethra. To get a urine culture, the genital area must be clean prior to collection, and then usually they catch a clean midstream of the urine, meaning the patient urinates and it's not collected, and then as they're urinating in the middle, you collect some, and then you do not collect it at the end of the urination. This is called a clean catch midstream, which most patients like to do because all they're doing is urinating into a cup. There is another way of doing it, and that is catheterization, where you put a catheter up the ureter and into the bladder, and then directly sample the urine in the bladder. The catheter reduces the risk of contamination, but the lower portion of the urethra does have bacteria in it, and as the urethra goes, the catheter goes through the lower portion of the urethra, it can take bacteria up into the bladder. An even better way, which the patients really hate, is suprapubic aspiration, where you put a needle directly into the bladder and then sample fluid directly from the bladder. Okay? Any question about the 3 ways we collect urine to test for a UTI? Oops, too far. All right, so once you obtain the urine, only 0.001 milliliter, or 1/1000th of a milliliter of urine, is placed on a non-selective media like blood agar plate using a special inoculation loop, which is shown here. Its size is to put 0.001 milliliters of urine on a blood agar plate. You streak it out like that and like that. In addition to growing the bacteria on a blood agar plate, which will allow everything to grow, It's also a good idea to put the urine on a McConkey plate or an eosine methylene blue plate, because these media only allow gram-negative bacilli to grow. And the reason for this is the most common cause of urinary tract infection is a coliform like E. coli. And so they will grow on the MAC and the eosine methylene plate, whereas the gram positives will not. And this shows you that this bacteria here growing on the MAC plate is gram negative, and it's also pink, so it's fermenting the sugar lactose. The interpretation of a positive urine culture is multifaceted. Factors that need to be considered is the type of collection. Voided versus catheterized specimen, meaning a catheterized specimen will have fewer contamination than a voided specimen. The gender of the patient, whether male or female. Generally, females have more bacteria in the urine than males. The number of colonies per milliliter of urine, and the presence of polymicrobial growth. If there's multiple organisms isolated, it may indicate poor collection, and you're not getting as much of the bacteria causing the urinary tract infection as you would like. All right, so once we get the bacteria growing, we can then run different tests to help identify what is causing the urinary tract infection. You can run the coagulase test, and it is especially used for differentiating Staphylococcus aureus from other Staphylococcus species. Staphylococcus aureus is coagulase positive. Other staphylococcal species are coagulase negative. Coagulase is a enzyme that converts blood plasma protein into fibrin, which causes a clot to form around bacteria in the blood. And Staphylococcus aureus uses this to be more pathogenic. And the reason is that if you put a clot around the bacteria when they're low in number, the immune system has a harder time removing the bacteria from the blood when they're low in number, which is the easiest time. And so Staphylococcus aureus puts a clot around itself, and it makes it harder for the immune system to remove the bacteria from the blood. And this is showing you fibrinogen, and coagulase creates a fibrin clot, which is seen right here. This is clotting, this is not clotting. So this would be coagulase positive, this would be coagulase negative. And there are two possible tests. This will be the one I'll give to you in the unknown. You could also do it in the slide test. This is coagulase positive, there's clumping, and that's coagulase negative. The most common bacteria isolated from urine cultures is E. coli, which is in the family Enterobacteriaceae. And there are specific biochemical tests that you can use to identify this family. For example, all of this family are gram-negative bacilli. They all ferment glucose. There is another gram-negative bacilli, though, that's in another family. It's in the Pseudomonaceae family, and Pseudomonas aeruginosa is a bacteria that can cause a UTI. However, Pseudomonas aeruginosa is an aerobe. We didn't mention that, but Enterobacteriaceae are facultative anaerobes. And then Pseudomonas cannot ferment glucose, whereas the Enterobacteriaceae can. And then there's one other difference too, and that is the Pseudomonas will test positive in the oxidase test. It'll turn blue in the oxidase test, and the Enterobacteriaceae are negative in the oxidase test. All right, any question about any of that? That's it for the lab manual. Remember, don't write your answers in the lab manual. Write them in the worksheet. You're not physically going to be performing any of these activities. However, you will be responsible for knowing how to perform them. We do have video clips on running the catalase test, the coagulase test, and the oxidase test. Once you watch those videos, then answer these questions. Okay? Most of these questions can come from the lab manual, meaning answered from the lab manual, or from the videos. There was one question that cannot be answered. I don't have it here. Let me see if I can find it. And I've got it scratched out. Let me show it to you here. Yeah, 8C. You don't need to answer 8C. All right, so answer your answers in the worksheet, and it's not really too bad. The hardest part will be identifying the gram reaction and cell morphology of these bacteria. So tell me, is Staphylococcus saprophyticus gram positive or gram negative? And tell me its shape. Shape means, is it a cocci, is it a bacilli, or maybe a coccobacillus? Okay? All right, any questions? If not, I will be here until, let's say, 7:45 today to answer questions.